Here we go. Okay. Oh yes, this week's study tip is just about remembering to practice. And so, if you were going to run a race, you would practice by running often, regularly. You would also do other types of training and diet and, et cetera, et cetera. And so when you're preparing for exams or quizzes, you want to practice answering questions like the types of questions you'll have on your exam. So for us in our class, it's multiple choice. So you want to use the multiple choice questions that you find, for instance, in your lecture textbook. At the end of each chapter, there's practice questions. You could practice re-answering our homework questions from the discussion homework. You could also look at those quiz questions from Lapster, and all of those are ways to practice. And that's after you've done your studying, you use these practice questions to see what you retained and what you understood. Never start with the questions first. That defeats the purpose. Always put your notes away after you've studied and practice the questions. If you had a writing on an exam, like when we're in person, I usually have one or two free response questions. And so then I would encourage you to write full answers out, just like you would on your exam. Of course, that doesn't apply to us. Okay. Introduction to the immune system. So immunity, as you already probably know, includes all the structures and all the processes that provide defense against pathogens. And as you already know, a pathogen is any disease-causing organism or agent. We have to add or agent because the viruses are not living. Everything else, bacteria, fungi, protozoa, those are all living and can, if they're a pathogen, they can cause disease. Okay, so I want to remind you that, microbiology focuses on diseases much more than we focus on any other application. And that gives you a really skewed perspective. Less than 1% of all bacteria in the world cause disease. All the rest of it's good. So we have a lot of gut microbiota, microbiota that live on our skin, right? So we have all these bacterial cells on us and in us that we need in order to survive. They make vitamins for us. They help break down certain foods. They protect us from pathogenic organisms colonizing us to some extent. So they're very important. And then, of course, those that are not on us or in us would be environmental microbes, which have lots of very important ecological roles, such as decomposing, returning nutrients into the soil, taking nitrogen out of the air, as we saw in our eutrophication lobster. You saw a little bit of environmental science there. Okay, symbionts, there's organisms that cannot survive without their bacterial or fungal partners. So hopefully this helps remind you that, most bacteria, most microorganisms are actually good, helpful, friendly. Okay, so now back to the pathogens. Our immune system is divided into two systems: innate, which is non-specific, and adaptive, which is highly specific. So the immune system is composed of white blood cells, abbreviated WBC. And all of these white blood cells originate from the bone marrow. So you see the red bone marrow here. And the special stem cells, hematopoietic stem cells, are the stem cells that all blood cells come from. So in this image, you'll see red blood cells here and platelets, which are not part of the immune system. But everything else that we see on this image is part of the immune system. So from this stem cell, you can become a lymphoid progenitor cell or a myeloid progenitor cell. If it's lymphoid, you're going to produce one of the 3 types of adaptive immune cells, and we'll talk about that later. If you're from a myeloid progenitor cell, you're going to produce one of the several types of innate immune cells, eosinophils, basophils. Monocytes can further differentiate into either dendritic cells or macrophage. We'll talk about these in a moment. And then you have neutrophils. And neutrophils, you could put next to this as number 1. They are the most numerous of all of the white blood cells in your body. Okay. We will see this image again when we start talking about the adaptive immune system, just to remind ourselves. This, memorize this, draw this out. This is a very, very useful tool. Okay. So now let's talk about the innate or non-specific immune system. The first line of defense, which is really prevention of even trying to not even let bacteria get into the body. These would be chemical and physical barriers. And we'll talk about those in a little bit more detail in a second. But let's say you get pathogen gets past the first line, then you have the second line of defense, which will be non-specific immune responses. And this involves the phagocytic cells, so neutrophils, monocytes, right? And then natural killer cells can respond as well. So the different cells of the innate immune system. And then these cells can release, and other cells as regular body cells as well, can release interferons, which are signals. We'll talk about those. You can have a very localized reaction, right? So say you prick your finger on something, or you cut yourself somewhere, you'll experience local inflammation, right? You don't need to have an increase in immune cells in your leg if it's your hand that you injured, right? So localized inflammation to take care of a localized problem. Now, if you don't get this under control, or you have a large enough infection, so even if it stays local, if it becomes a big enough problem, systemic inflammation as well, which would be a fever. And then lastly, the complement system, which is not strictly innate or adaptive. It's kind of this other system that works with or complements both innate and adaptive. And we'll talk about that last. And then once we talk about all this, then we'll move on to the adaptive. We're not going to go into quite as much detail for the adaptive immune system. And then that will be the first lobster. And then for the second lobster, it's just antibodies. It will also be a little bit easier, quicker, not easier. It's all pretty much the same level of difficulty, but just faster, less information. Okay, so hang in there for the innate. Once you get through the innate immune system, I'll move pretty quickly. Okay, are there any questions before I move on? All right. So innate immunity, remember, is non-specific, so you're never talking about antibodies. You do not make or use antibodies when you're talking about the innate immune system. The only exception would be the complement system. The complement system does have a pathway that uses antibodies, and it can also promote the adaptive immune system as well. But otherwise, nothing else in the innate immune system will ever talk about antibodies. Okay, automatic responses to an invader. It doesn't know what the invader, who it is. It doesn't know, oh, E. coli is here. right? Or influenza virus, right? It doesn't know what the specific invader is. Our body can usually distinguish between bacterial and viral infections for the innate immune responses, and that's pretty much it. So it's going to be very general response, okay? And these responses come from, of course, our genes. Everything in our body comes from genes. And so we inherit these genes for innate immunity. And again, that's going to just give these generic responses. And mainly, it's the body utilizing our self tags. So we know what is us. And then by default, anything that doesn't have our self tag would be foreign. And self tags, let's go ahead and make a note here. So all of your cells of your body have MHC class 1. And so you could put somatic or regular cells, and then your white blood cells will have MHC2. And we probably won't even talk about MHC2, but you will see MHC1 today. So whenever we say self-tag, we're talking about MHC1. Unless we're talking about a white blood cell, then we're talking about MHC2. Okay. Arrow. Okay. So foreign cells that are not us, are going to have different markers, different types of proteins, carbohydrates, all these different molecules, which are called antigens. And we can classify this whole group of pathogenic antigens as pathogen-associated molecular patterns. So PAMPs, pathogen-associated molecular patterns. And so that's all the different types of antigens. This is going to include your surface antigens. This is going to include things like DNA, RNA, different types of carbohydrates. For example, bacteria all have peptidoglycan. We never have peptidoglycan in any of our cells of the body. So that's easy to recognize. If we can get access to it, we will definitely respond to it as being a foreign antigen. So our cells of our body have toll-like receptors that recognize PAMPs. So again, our body has TLRs. The pathogen has the PAMP, and if any of our TLRs come into contact with a PAMP, that's going to sound the alarm system. That's going to trigger an immune response. Okay. So a physical barrier would be our skin. Our skin is definitely taken for granted. It's an amazing organ. It's our largest organ. It replaces itself every few months. It protects us in multiple ways. It's dry. It's fairly impenetrable, and it sheds, right? So even if a pathogen manages to hang on to the skin and start to colonize the skin's going to shed. So unless it's able to get further down, like maybe being sneaky and getting into a hair follicle, otherwise it's going to fall off when our skin falls off. Okay. Mucous membranes, they create mucus, and that mucus can trap microbes, and then it will be physically removed as the mucus is removed. I didn't realize I wrote over this, so I'm going to erase these notes. Okay. Pen. Okay, so physical removal could be just removing mucus from the mucous membrane. So an example of that would be the ciliary escalator. So your lungs are covered in mucus, and in the respiratory system. But especially in the lungs, we have cilia that constantly propel the mucus up and out, and then you swallow it. And it's about a liter, I believe it's about a liter of mucus every day that we produce, and the majority of that's going to be in the lungs. But of course, you know, your nose, your mouth, your eyes, and your reproductive organs and such. Okay. Of course, vomiting would be a physical removal. We don't typically vomit, so that's kind of a unique situation. Urine. Urine, a lot of people have a misconception that urine is sterile. It's not sterile. I don't know why people say that, but it does help remove pathogens. Okay, so perhaps urine as it's filling into the bladder is sterile. Granted that you don't have an infection of the kidney, then your urine would be sterile. But once your urine leaves the body, it's exposed to your urethra, which is covered in bacteria. So then if you were to collect urine in a urine sample, you would not expect it to be sterile at that point. Anyways, urine, you need to regularly urinate so that you can remove the bacteria from the urethra before it moves into the bladder. Once it reaches the bladder, then it can establish an infection. And of course, that's dangerous because it could further move up. And if it gets to the kidneys, a kidney infection can potentially be life-threatening, right? So kidneys are a vital organ. So that's important. Okay, next. Chemical barriers, the enzymes. We make enzymes that break lyse open bacteria. We also have other antimicrobial peptides that can harm bacteria. And then we can also use pH. So we can make, like for our stomach acid, it's highly acidic. It's going to kill most any microbes that you consume. We also have enzymes in our tears, in our sweat, our saliva, and our mucus. So mucus and urine, all these that can be involved in physical removal also have chemical components in them. So it's not always easy to completely differentiate between your physical and chemical barriers. But of course, chemicals are anything that is going to directly attack, break down the microbe, whereas a physical barrier would be preventing it from getting entry and physically removing it. Okay. Now, we also have phagocytes that search for and remove pathogens. As I mentioned before, neutrophils are the most abundant, and they're typically first on scene because they're most abundant. And they usually, once they encounter a pathogen, they release some cytokines that will recruit more white blood cells to the scene, including monocytes. Monocytes can also sometimes be the first one to encounter a pathogen because we have monocytes that are like what we call resident monocytes that are kind of always on the lookout in that particular tissue. And monocytes, once they come in contact with a pathogen, will either differentiate into a macrophage or a dendritic cell. And macrophages will they're like vacuum cleaners, right? They're going to clear up cellular debris. If there's a pathogen like here, it's going to phagocytize it, eat it up, destroy it, kill it, get rid of it. In the process, when this happens, as it's being a little vacuum cleaner, when it does come to contact with bacteria, like in this picture, it's going to release some cytokines, specifically called chemokines, just to let the other cells know that it did indeed encounter some bacteria, and we could have a possible infection. So that's going to recruit more white blood cells to the scene to make sure that everything's good and clear. If it becomes a dendritic cell instead, this is useful because dendritic cells are what we call antigen presenting cells. They present antigen to the adaptive immune system. So we call dendritic cells the middleman between innate and adaptive, because though they are not doing anything specific, right? They don't know when they take up a pathogen, they don't know what kind of pathogen it is. They just know to display the antigens of that pathogen and go find a B or T cell and present it. And of course, if it presents to a B cell, then you're going to get antibodies. If it presents to a T cell, you can get cell-mediated immunity, such as cytotoxic T cells, or a helper T cell to help the B cells make antibodies. Okay. So what is phagocytosis? I said it'll take it up and eat it and destroy it, and that's kind of true. So chemotaxis is when the phagocyte moves toward some cue that attracts it, some attractant. And then, of course, it has to adhere or bind to the pathogen. So right here, we have adherence of the bacteria to the phagocyte, right? So the phagocyte grabs on, and you see here, like it really does, this white blood cell really does grab on and eat up these bacteria. And then once it grabs onto it, it's going to bring it into itself. So it's going to create a phagosome. It's going to invaginate and bring it into the cell, contained safely in a vesicle. Okay, then it's going to digest it. So you're going to get, you're going to get, did I undo this? Yes, I did. Sorry. Okay, so once you have the phagosome, then this lysosome with the digestive enzymes is going to fuse to the phagosome, which of course we just call the phagolysosome. We're lazy, we just put the two names together. And then that's going to digest it. It's going to break it up. And then you get some waste products that eventually can be released. And these are going to be very, very small, because phagocytes are already vacuuming up debris, right? So it doesn't want to just make more debris. Anyways, that's the process of phagocytosis. It's taking up bacteria or viruses or anything else that's small enough for it to take in and digest. Okay. You may have learned more detail than this for lecture, but this is as much detail as you need to know for lab. So just a reminder, different phagocytic cells. We talked about neutrophils, the most abundant type of white blood cell in the entire immune system. They are phagocytes, so little cellular vacuum cleaners, right? We have monocytes, and we talked about monocytes they circulate in the blood, and we also have some tissue that live in the tissue, and monocytes can be turned into macrophages, or they can turn into dendritic cells. The purpose of a dendritic cell is to present antigen, which is why you don't see it on this chart, because this is talking about phagocytes who their primary job is to be a phagocyte and just clean up messes, right? So if you're a resident macrophage, you could live in a particular tissue like the spleen, the lymph node, bone marrow. And then of course we've given specific names to macrophages that live in places like the liver, the lungs, or the central nervous system, right? So microglia, you can find those in the brain, which is pretty much the only immune cell that you find in the brain. We have to be very, very careful with the central nervous system. In the lungs are constantly being exposed to air, all sorts of junk in the air. And so we have alveolar macrophages, which are like vacuum cleaners for the lungs. Another reason why you shouldn't smoke. You already know about damaging the cilium, getting a buildup of mucus in the lungs, having chronic inflammation of the lungs, having COPD. But you also don't want to damage your alveolar macrophages, and you don't want to add to their job. If they're so busy trying to clean up debris from your smoking, they are less likely to be able to catch pathogens, right? So another reason not to smoke. All right. Another type of white blood cell of the innate immune system are natural killer cells. These will just look at every cell they come into contact with and make sure that they have a self tag, MHC1. If a cell is missing a self tag, then the natural killer cell is going to insert granules, which contain perforin, which poke holes in the cell and cause it to lyse open. So the natural killer cell will go around and kill any cell that is missing a self tag. So in this way, it is non-specific. It doesn't care what the reason is. It doesn't care if it's a particular pathogen. It might even be a cancerous or precancerous cell, or hopefully not, but a mismatched transplant tissue can also cause this response. So cells mutate all the time. They are constantly getting exposed to different chemicals and radiation, right? All sorts of processes that damage cells. And so every day our body must find and get rid of these mutated cells before they turn into actual cancer. So medical cancer is different than like cellular level. So like as a biologist, when I say a cancer cell, I mean a cell that's replicating or behaving in a way that it shouldn't. So it's just uncontrolled. It's a rebel. It's not behaving, right? That's not the same as having actual like medical cancer where you have a malignant, tumor. Okay. The idea, the hope is that our immune system is working well enough to prevent actual medical cancer from ever happening. So this is another reason to take care of yourself, to, eat your fruits and vegetables, get exercise, drink plenty of water, get plenty of sleep. All those things will help your immune system to be able to do its job and prevent cancer. Okay. So interferon or polypeptides can be released from cells when they become infected, basically to warn their neighbors. It's too late for me, but save yourself. So a cell, it becomes infected and there's like some foreign nucleic acid, right? It's like, what is this DNA? A virus, bacteria? I don't know, but clearly I'm infected. So it's going to release these signals called interferon to tell the neighboring cells, watch out, we have an infection. And sometimes that's a virus. So if it releases alpha or beta interferons, it's going to release a virus signal, and it's going to help its neighbors to resist. It's going to create particular enzymes that block the virus from being able to get inside and replicate. If it's a different type, like interferon gamma, then this is more like intracellular infections of bacteria or even cancer. Okay, local inflammation. So let's say you accidentally cut yourself. You're going to have tissue damage those damaged cells are going to release some histamine. Also, your resident phagocytes, your mast cells, if they come into contact with a foreign particle, they're going to release chemokines as well. Or when you damage your tissue, if you damage those, they also release. If the phagocyte or mast cell gets damaged, it will also release chemokines. Mast cells in particular will release histamine, and that serves the purpose of making the capillary cells kind of loosen up those tight junctions so that white blood cells can squeeze through and get into the tissue. The process of white blood cells moving from the capillary to the tissue is called diapedesis. There's many steps involved in that. And since this is lab, I'm not going to go over this. It's not in the simulation, so I don't need to give you the specifics of diapedesis. If you want to talk about it later, just ask. I'd be happy to go over it with you. Okay, so in addition to the histamine increasing capillary permeability so that white blood cells can squeeze through and get into the tissue, you're also going to have localized vasodilation. So the blood vessels become larger so that more blood can flow through them. And that's going to help bring more white blood cells to the area. And then, of course, they will need to go through diapedesis to get into the tissue. When you have localized inflammation, you're going to have redness, warmth, and it's going to be warmer than the rest of the areas of the body. Usually there's plenty of pain involved. And that pain can be due to both the damaged tissue, but also the swelling of all this extra blood flow. And then once your white blood cells have started working on fighting the infection, you can get the formation of pus. Okay, so that's localized inflammation. Let's say this gets out of hand or starts to spread to other areas, then you're going to have systemic inflammation, and that is fever. So general non-specific response to any infection. One of the chemicals involved is called pyrogen. This is going to reset your hypothalamus to a higher temperature, and that's going to tell your body that you're too cold because you've been keeping it 37 degrees Celsius. Now it's set to 38, and your skeletal muscles are going to start shivering and building up extra heat very rapidly. So this is the part of the fever where you feel cold and you want to bury yourself under covers and warm up, because your body really does believe that you're too cold because the thermostat has been reset. Okay. Once the fever starts to break, you have lowered the amount of pyrogens, you no longer need the fever, so now your thermostat gets reset, then you would experience the feeling of being way too hot and sweating and cooling off. Okay, I didn't include that in the slide because that's the breaking of a fever, and we're talking just about fever in general. So in addition to increasing skeletal movement to produce heat, you're also going to vasoconstrict all of your extremities to keep all the blood as close to your internal organs as possible. And why? Why does the body have this system of fever? Well, it can be very beneficial in helping you fight an infection. So some pathogens, they don't work very well at the higher temperature. For instance, pathogens that normally live in the environment and then cause infection in us, we are much warmer than the environment. So we might be at the very upper end of their limit of what they can tolerate for heat. And having a fever can slow them down or even kill them if we exceed the temperature that they can, their enzymes can work at, right? But even if our fever doesn't affect the pathogen, it still is really beneficial because it's going to increase our immune system's going to go on to overdrive. We're going to have extra proliferation and activity of our T and B cells for the adaptive immune system. It's going to increase the rate of tissue repair and the rate of interferon production. So different parts of the innate and adaptive immune system will benefit from a fever. The fever in and of itself is innate. It's non-specific response. A lot of people are afraid of fevers. You shouldn't be, but you should always monitor a fever. You should have a thermometer handy. You should know what your normal temperature is. That way you'll know when you have a fever. And then you should monitor how high the fever gets. You should also, monitor your heart rate, make sure that you stay hydrated because the fever can cause dehydration. It can cause rapid heart rate. If you get dehydrated, that's going to increase your heart rate even more. And of course, if the temperature gets way too high, then you can start to have hallucinations, right? You can become delirious. If it gets any higher than that, it can actually cause permanent brain damage. And of course, ultimately, if you get so hot that you start to denature your own proteins, then you can die. In the notes, I included the specific temperatures for infants, children, and adults. Of course, if you ever, if you or a family member has a fever, you can always just call any doctor's office or hospital and get a nurse to help you decipher if it's time to see the doctor or not. Usually my personal rule of thumb is if it's over 102 degrees, I'll take some Tylenol. If it's 103 or more, even after taking something like Tylenol, then it's time to go to the doctor. But otherwise, you do want to let the fever do its job, right? Okay. Complement system, as I mentioned at the beginning, it's not really innate or adaptive, but we're including it in the innate immune system because you have these complement proteins that are always circulating in the bloodstream, and they can come into contact with pathogens or antibodies that are bound to pathogen, and that can, what we say, cleave the complement protein and start the cascade of cleaving these proteins and forming new products. I'm not asking you to know any of the specific proteins of the complement system. You may need to know that for lecture, but for lab, you do not. For lab, I just want you to know the concept of it. So you have your simulation actually tells you about 3 pathways. I really only care about the alternative and classical pathway. And again, keeping it very general, alternative pathway is when the complement actually attaches to the pathogen directly. And cleaves, it splits, and it starts the complement cascade. That's non-specific. The complement protein doesn't know what kind of pathogen it is. It just reacted, just chemical reaction. Whereas the classical pathway, it's responding to antibodies being attached to a pathogen. So here you have these 2 antibodies side by side, and then you can get a complement protein complex that binds to that, and then that causes a cleavage and releases. I blocked out all the specific names because it's overwhelming when you see all the different C3, C4, C5, right, and all the different names of the proteins. I don't personally care about that for an introductory level class like this. So you get this cleavage of the complement protein, and that it starts the cascade where you get this protein and these proteins, right? And so the ones that get released into the, like, they're not being bound into the membrane, but they're being released into its environment, usually the bloodstream. That's going to go and enhance things like inflammation when it binds into the membrane, it can lead to the formation of membrane attack complex, which will poke a hole and lyse open the bacterial cell. This is like a 2-minute video. It's really good for giving you a very general overview of the complement system. So please watch that video. Okay, so again, you have these 2 main pathways, alternative and classical. As the complement protein, which again, the complement protein is always in circulation in the blood. And if it becomes activated, it cleaves, and you get a cascade of all of these different cleavages of different proteins of complement proteins. And some of those are going to increase inflammation and phagocytosis, and others are going to embed into the membrane of the pathogen directly and poke a hole and cause it to lyse open. Okay, so that is the end of the innate immune system. Yay! Now we're at the adaptive immune system. Personally, I find the innate immune system super fascinating and lots of fun, but I know it's also a lot of information. So make sure you give yourself plenty of time to study this. Okay, adaptive immunity, we're also going to keep it pretty entry level. So often the adaptive and the innate immune systems are working together. They're working simultaneously as well, right? So sometimes we don't see the effects of the adaptive immune system for 7 to 10 days, while the innate is working. You don't even necessarily feel the innate immune system working unless you get the fever, for example, right? And once the adaptive immune system's in full-blown proliferation mode, you can feel those symptoms of that as tiredness, weakness, and maybe even a slight fever, right? Okay, so anyways, when we talk about the adaptive immune system, we are now talking about lymphocytes. So the right side of this image that we looked at earlier, where you have the lymphoid progenitor cell, and that can form T cells, B cells, or natural killer cells. And so these are all lymphatic cells. Okay, B cells can then form plasma cells, which secrete lots of antibodies. Or, and you should draw another arrow, you could put memory cell, which is a huge component of how we get long-term immunity. T cells can also form memory cells, and T cells are involved in cell-mediated immunity. So we have 2 types of immunity within the adaptive immune system. Okay, so in the bone marrow, we had the stem cell, and it can make the lymphoid cells, or the stem cell could make the myelocytes that we looked at for the innate immune system. So just trying to get your perspective back here. Now we're back in the bone marrow, and we're looking at the lymphoid, and we call it a common lymphoid progenitor because this can make T cells, and T cells go to the thymus to mature. It could make natural killer cells, which migrate to the lymph node to mature. And it can also make B cells, which stay in the bone marrow to mature. And you'll notice that these cells have to go through central tolerance testing before they are released to the lymph node, where they go through peripheral tolerance testing. So central tolerance testing is making sure that the cells are not going to react with cells of the body, okay? So preventing autoimmune disease. Whereas peripheral tolerance testing is going to make sure that these cells are not going to overreact and cause such a big immune response that you go into shock or possibly die, Okay, so our immune system is incredibly powerful. It's powerful enough to kill ourselves. So we have to control our immune response so that we don't harm ourselves or we limit the harm to ourselves. Okay, so peripheral testing is to make sure that we're not going to have overactive cells that give us way too strong of an immune response. All of the peripheral tolerance testing, as you can see in this image, is taking place in the lymph node. Whereas the central tolerance testing just depends on where the cell first starts its maturation process. If it survives both of these tests, then it gets to survive and stay in the lymph node. Some of these will pass through the lymphatic system, but most of your white blood cells lymphatic T and B cells and natural killer cells. Most of them are going to stay in lymph nodes and scan the lymph fluid for any antigens. You do have small amounts that circulate even in the blood, right? So you know that you can have a blood test taken and you can have a differential blood count where you can see how many red blood cells you have and how many white blood cells you have. Furthermore, the breakdown of what types of white blood cells. And since we know what's normal or average, we can use that information to see if someone has an infection. If they have way more white blood cells than usual, then you know that they're currently fighting an infection. Or there's another issue like autoimmune disorder. Okay, so in the homework, I want you to tell me what the central tolerance test is. You'll read about this in the theory tabs and take notes because I went over this in a little bit vaguely. You'll want to gather a little bit more detail for central and peripheral tolerance testing. Okay, that's one of your homework questions. What can happen if a cell doesn't get caught by this test and is allowed to move forward, right? So what is the test really preventing? I briefly went over that. Okay, so now let's talk about antibodies. So if your B cells get activated, they will make antibodies. The very first antibodies to get made in response to infection, well, if it's a viral or even bacterial antigen that is in your mucous membranes, the very first antigen you'll see is actually IgA. But if it's something like respiratory or other area, you're going to see the IgM first. And so you'll notice that the IgA and the IgM are kind of short-lived, and the IgG, which takes longer for your body to produce, will eventually get produced to a much greater amount than the first ones on scene. So when you do a blood test on someone, if you see elevated IgM levels, then that would indicate that they're fighting an infection or they just recently fought an infection. But IgG would indicate that they have long-term immunity, and that could have been from a vaccination or a past infection. Okay, but we wouldn't look at IgG and say, oh, you're infected, because that took a while for the IgG to really get to these levels. And probably you already feel better and have already cleared your infection by the time you get to these IgG levels up here. Okay. Now notice this isn't marked with specific days because this can vary, right? So typically 7 to 10 days for the IgA, IgG, IgM, and then another few days for IgG, and it continues to ramp up. And then you're pretty far out by the time you get to this upper branch. So let's say if this is 7, 8, 9, 10, so you're looking at day like 12, around day 12, and then farther out. So yeah, most people, when they are ill, their symptoms subside in 1 to 2 weeks. And typically that also means they're not contagious. Of course, that varies depending on the pathogen. Some pathogens you actually continue to shed at a very high number even after you start to feel better. Others, as you feel better, you become very less and less contagious. So it's kind of hard to give these very general discussions when we have kind of a range of behavior depending on the pathogen. But in general, we can use this pattern. IgM is first, IgG is second. IgG is associated with long-term immunity. This will continue and continue, whereas IgM, you just made like a batch and then it wears out. Okay. Another thing to think about is what I was talking about with how long it takes to make these antibodies. That's the primary response. That's the very first time you get exposed to the antigen, whether that be a vaccine or an actual infection. It takes about a week to start making antibodies, and about, this is saying for the most, it takes even 4. So that would be the peak of your IgG. And then your IgG will stay at a little bit lower, but it will always stay in your, you'll constantly make that. And that's why it's long-term immunity. Okay, so that's the primary response. It takes time. The secondary response, if you ever encounter that same pathogen again, you are going to immediately, day 1, day 2, immediately start secreting huge amounts of antibody because you have those memory B cells, right? You also have memory T cells, which immediately get to work as well. And you will never even experience a single symptom. You'll have no idea because all the hard work had already been done, and your memory response is able to just get to work, and it really gets to work. Look how beautiful that is. Such a high amount. And that lasts quite a while. I mean, you're 5 weeks out and you still have more antibodies than you did originally. So hopefully the function of the immune system has really impressed you. Okay, and you can see why it's so important to get your vaccines. Okay, so active immunity is when your bodies produce antibody yourself, right? So you're exposed to a pathogen and you get an army of B cells and T cells for cell-mediated, and both B cells and T cells will make memory cells, and that's going to give you a super rapid response if you ever come in contact with that pathogen again. And that's how we get our long-term immunity. This could be through natural exposure, like getting sick, catching a cold or influenza virus, etc. Or it can be artificial, which is from a vaccine. And so a little note about vaccine is the vaccine's exposing you to antigen, but it is not giving you any risk of infection. And how it does this, well, how do we choose what to make a vaccine of? There's lots of different pathogens. Well, we target the pathogens that affect a large amount of people, and either that are causing death, high death rate, or are causing a lot of complications. So, this is another time you could talk about COVID a little bit, is that COVID's not necessarily the most lethal virus in the world, right? It's not killing people like at the same amount as the Black Plague did, but we're seeing a lot of people who are ill for a very long amount of time. Their recovery is slow, and they were even seeing people with further complications, not just having the initial illness that lasts a couple weeks, but some people are having weird nerve disorders and cognition. We're just seeing a lot of side effects as this. As we collect more and more data and analyze the data, we're seeing that COVID is causing a lot of problems, not just mortality, right? But even still, the mortality is definitely high enough to warrant making a vaccine. So that's how we target, that's how we decide what vaccines get made, is what do we need protection from the most? What is affecting the community either through death or through illness and complications? And so that's another good example to use would be polio. You might not even know what polio is because we've had the vaccine for it for so long now, and it was almost eradicated from the world, but there have been some hot spots where it still is around, but it's not. I don't think we have any cases in the U.S. But polio didn't kill people very often, but it caused a lot of people to become paralyzed in one or more limbs. That could be a leg, it could be an arm. But imagine getting what seemed like a cold and then having your muscles just deteriorate and no longer being able to have use of that limb. Some people were unfortunate enough to have it happen to more than one limb, right? Be wheelchair bound or or whatnot. So polio was just so awful, and it was so contagious. It pretty much everyone would get it, and because everyone had it, and a small percent of a lot of people is a lot of people, right? So yeah, that's how we choose our vaccines. They are so valuable. They're one of the most valuable medical resources that we have, much more valuable than antibiotics. And you know how glorious those are, right? Okay, so why people are so afraid of vaccines is because they don't understand them, and there's been a lot of misinformation spread about them. But I want to reassure you that even if you experience a slight fever, tiredness, soreness, you did not get infected. It's just the activation of the immune system making all those antibodies for you, protecting you. And it's so incredibly rare, but there are some people that are allergic to certain ingredients, and then they will be their medical records would be marked and they would not be given any more vaccines. This is another reason why everyone who can should get vaccinated, to protect those people who can't. Some people cannot get vaccines because they have immune diseases or other reasons besides this, but this would be a very small subset of people who are not able to get preservatives, in addition to like babies who have not had their vaccinations yet. They slowly get different vaccinations over time. So everyone needs to get vaccinated to provide herd immunity to those people who cannot receive vaccinations. Lastly, I want to address that there is absolutely no way that vaccines can cause autism. Autism is a genetic disease, and vaccines cannot interact with your DNA. They do not interact with your DNA. Vaccines simply activate your immune system so that your B and T cells make memory cells. So if they ever come in contact with that particular antigen again, they are ready to protect you. Okay? All right. Passive immunity would be receiving antibodies from another person or even an animal. We've seen this in COVID. We've seen this also when we had the Ebola outbreak. Man, that might have been like 2010. It's been a while, but basically when we don't have a particular treatment for a disease, right? We don't have an antiviral or or a proper antibiotic. We don't have anything to help a person that's fighting for their life. We can try finding and purifying antibodies and giving those to the person to help them fight the infection. We also see this as a natural process between mothers and babies. The mother can pass her antibodies to the fetus because some of them are small enough to cross the placenta. And then also during breastfeeding, some antibodies are passed that way as well. And of course, this is temporary. Once those antibodies are used or fall apart, that's it, no more. So the recipient will not have the ability to make more of these antibodies because they never made them in the first place. In order to make antibodies, you have to have the immune response. So this is temporary, and it can be natural or it can be artificial, right? Receiving it from another person through an IV in the hospital would be artificial. Receiving it from your mother, of course, would be natural. Cell-mediated immunity is using T cells. And we have lots of types of T cells, but we're going to talk about 4 main branches of T cells. Regulatory T cells, their job is to prevent hyper or overstimulation of the immune system, basically to prevent harm to yourself. Helper T cells are activated by antigen presenting cells like the dendritic cells we talked about earlier. And then once the T cell is activated, it will go tell either a B cell or a cytotoxic T cell. And if that B cell or T cell also encountered that antigen, then that's going to be, it's basically saying, hey, I found this antigen. And the B cell says, yeah, me too. And then they both just proliferate and start doing their job very quickly so that you can fight this infection. Okay. So this is why it can take time, for antibodies to get made, because the B cell has to come in contact with the antigen and a helper T cell has to come into contact with the antigen. And then both the helper T cell and the B cell have to both talk to each other and say, yes, we both found this antigen. You are correct. This is not a fluke. This is not a drill. You have a real invader. And go, proliferate, make lots of clones of yourself, and then lots of antibodies. And from this proliferation of the B cells, some of those become plasma cells, which their only job is to secrete antibody, and some of the B cells will become memory cells, and their job is to stick around and give you long-term immunity. If you ever encounter that antigen again, they will immediately secrete lots and lots of antibodies. So the memory cells will become plasma cells if they encounter their antigen in the future. Cytotoxic T cells, on the other hand, are also activated by helper T cells, activated helper T cells, and they will seek out, hunt and kill the specific pathogen. And you will also make some memory cytotoxic T cells as well. Another group of T cells would just be naive. They have not encountered any antigen yet. They're not active. They're just sitting and waiting. And then, of course, there's the cytotoxic T cells that I talked about here already. They kill infected or cancerous cells. So it's kind of similar to the natural killer cells that we talked about, but this time they're specifically looking for antigen that's being displayed on the MHC1 tag. So originally I put with odd MHC tags, but we should be more specific than that. So they are looking for and killing cells that are displaying antigen on the MHC1 tag. So MHC1, remember, is a self tag, and they've modified their self tag with some additional like antigens saying I'm infected or I'm cancerous, right? And then the cytotoxic T cell will kill it. All right. So that is cell-mediated immunity. And then this was what I was saying, how natural killer and cytotoxic T cells seem like they're the same, but remember, natural killer is part of the innate immune system, and it's only going to kill cells that are completely missing an MHC1 tag, whereas the cytotoxic T cells are part of our adaptive immune system. It's targeted killing, it's looking for specific antigens, or an MHC1 that's displaying the specific antigen on it. Okay, now that was the first Labster, and that was long because I wanted to give you a thorough review or introduction to the immune system as a whole. A lot of this information you will use in the simulation, but the actual simulation itself is pretty simple. You just have one little experiment. It's nothing like some of our past Labsters. Okay, so real quick, the antibody lab. The antibody lab I really liked. I thought it was really good and to the point. All right, so let's fill in these blanks. See if you're still paying attention. Okay, so do you know the part of the antigen that binds to the antibody? The antigen's epitope will bind to the antibody's paratope. Maybe I should write that. Oops, oh shoot. Here we go. Pen. The antigen's epitope it's hard to write with a mouse. Okay, the antigen's epitope will bind to the antibody's paratope. Okay, sorry if you can't read that. This is also in the Labster, so it's okay if you're not getting it right now. Sometimes the paratope is also called the antigen binding site. So the part of the antibody that binds antigen, right? The part on the antibody that binds the antigen is the paratope, but because of its function is where the antigen binds, sometimes it's just called antigen binding site. Okay, next. Antigens can be lots of different things, lots of different types of molecules. So it's really anything that causes an immune response. Okay, so this could be nucleic acid, DNA or RNA, could be carbohydrates that you do not find in human cells. So I gave you an example of that earlier, is like peptidoglycan is a carbohydrate that we don't have. So if we detect peptidoglycan, we know it's an invader. Proteins, there are certain proteins that are unique to microbes that we wouldn't have, right? So any molecule that's on a microbe and is not part of us can be an antigen. And that's going to evoke an immune response. And that can be innate and that can be adaptive. And also the complement system. Okay. Antibody structures. So I mentioned this this binding site or the paratope. So there's a little bit longer sequence is called the variable region, and this is where our body just makes version after version after version, making very small changes over and over again, trillions of times. And so we have this part of the antibody that's called the constant region that does not change, but the green part does change. And that's why it's called variable. ahead and erase that, all these extra marks. And so, sorry, I can only do one thing at a time. Pen. No, I didn't want the pen, I wanted the arrow. Okay, so an antibody is made up of 4 polypeptides, which we call the heavy chain, it's the long one, and the light chain is the short one. And then we have the same thing on the other side. These are held together by strong disulfide bridges or bonds. And so the light chain is held to the heavy chain through a disulfide bridge. And then also each heavy chain is held together by a disulfide bridge. And you can imagine these must be pretty darn strong if that's the only just 1, 2, 3 of those to hold the whole antibody together. And antibodies can last anywhere from 2 weeks to months in the body. So they are really strong, okay? The variable region is found at this end. This end is called the NH4 end. Because remember, this is an amino acid sequence, and amino acids have an NH4 end and a COOH end, right? So COOH is found at the other end. And for the heavy chain, that's way down here. For the light chain, it's going to be at the end of the light chain, okay? And then all of the NH4s are at the top, which are at the variable region, okay? Constant region is the whole rest of the antibody, which never changes. All of your antibodies will be identical for all the purple spots, okay? That's it for structure. Now, antibodies can stay as singular Ys that we just looked at. We have IgD, IgE, and IgG, which are these individual or monomer antibodies. We have IgA, which puts 2 together, uses a strong, I think it's another disulfide bridge, or it's a little bit more complicated. But anyways, you hold 2 of them together, and that makes a dimer 2. And only IgA, which is found in your mucous membranes, will have this shape. And then pentamers, IgM, remember the first, your first antibodies made in response to an infection. These are bound in a complex. So they're not actually the best at binding antigen, but because you have so many binding sites, that makes up for that. And we have a term for that where this has a high affinity. It's really good and strong at binding antigen. These aren't as strong, but you have so many of them that you have strength in numbers. That's called avidity, and we're going to see that in a moment. So I mentioned IgA is found in mucous membranes, saliva, tears, breast milk. IgD is actually part of a cell receptor if you have that. Not everyone, right? We'll have a discussion on blood types in a moment. But this can activate basophils and mast cells. IgE is mostly for extracellular pathogens like parasitic worms. They're way too big to get inside of a cell, so they will exist outside of the cell. Typically, they will either be in the blood or in the intestine. But if IgE gets, they also are found in allergic reactions, allergic reactions when the body identifies something as a pathogen, but it's not. So like pollen, pet dander, right? These are just inert molecules, but the body recognizes it as being foreign and attacks, right? Okay. IgG, we've talked about quite a bit. It's your long-term immunity. It's able to cross the placenta, so that's very important. And we'll have a discussion about that in a moment. And then, of course, IgM, already talked about, that's the first responder in an infection. So responsible for early stages of immunity. Okay, So I talked about affinity and avidity already, where we have certain antibodies such as IgG that are really good at binding antigen. So they have high affinity for antigen. This one, they really shouldn't have put all of the antigen here because that makes it look like it is high affinity, but it's really not. So we're actually going to take a pin and we're going to say, you know, every so often it doesn't actually catch an antigen because it's not the best. But that's okay because it has so many binding sites that it has what we call high avidity. It's going to still bind a lot of antigen overall because it has so many binding sites. So it's the sum of all of the affinities of multiple binding sites, right? Okay. Then while talking about this binding, there are 4 types of bonds that can happen between antigen and antibody. It could be a hydrogen bond, it can be van der Waals, which is like slight charges attracting. It can be stronger than that. It could be electrostatic charges where you have ions, which are a true positive or a true negative charge. And then hydrophobic interactions, difference between water and non-water soluble molecules. So those are the 4 types of weak bonds that can form between antibody and antigen. So that's important to keep in mind. These are not covalent bonds. It is possible to detach, right? Whereas when we were talking about those disulfide bridges, those are strong covalent bonds. You're not going to have those break naturally in the body. Okay, so we started mentioning blood types when we talked about IgD, and that's talking about a type of red blood cell marker called rhesus. So first, you know the ABO blood group A. Some people are blood type A. They express A antigen on their red blood cell. Some people are type B. They have a B antigen on their blood cell, and others have both. They're AB. They have both A and B antigens, and still others don't have either, and they're called O. Furthermore, you've heard positive or negative, which refers to an additional antigen called the rhesus factor, and also sometimes referred to as antigen D. Okay, so if somebody makes an antigen against, if somebody makes an antibody against the antigen, that would be an anti-D antibody. They could attack those blood cells. And indeed, this happens. So we have to be very careful and we have to decide if someone is A positive or A negative, B positive or B negative, and so on and so forth. So if you have the Rh antigen on your blood cells, you are said to be positive. And so you would couple that with this system here. So you could be A positive, you would also have an Rh antigen, or you could be A negative and it would just look like this, and so on and so forth. Now they're showing possible antibodies in the plasma. A person would not have these antibodies in their plasma unless they had at some point come into contact with the antigen. So here it's saying you can have anti-B antibodies in your plasma, and that's going to protect you against blood type B, right, or B antigen. You would never have anti-A if you're blood type A because that would attack yourself, right? So here's saying you have the A antigen, therefore you can only make the antibody B, anti-B. If you are blood type AB, you cannot make any antibodies without harming yourself because you have both of the antigens. And again, this chart does not include the Rh factor. So if I were you, I would make another, I don't know, maybe if you can print this and write it in, or if you have the ability to write, like you have a tablet or something, and you can try to add in the Rh factor. You would actually need to add some rows so that you could show A positive, A negative, B positive, B negative, AB positive, AB negative, and O positive and O negative. So there's really 1, 2, 3, 4, 5, 6, 7, 8 different possible blood types. Okay. Furthermore, the rhesus factor poses a real problem for mothers. If a mother is Rh negative, it should say, this must have got a little weird when I was writing this, but if the mother is Rh negative and she has more than one Rh positive child, she can have antibodies that attack the fetus. So the first child is fine because the exposure to the Rh happens during childbirth, during labor. That's when the mom gets exposed to the Rh factor for the first time. And so it's not until her second Rh positive child that she would even have the antibodies, right? But then sadly, if she does have those antibodies, then it will attack the next Rh positive child, and it'll attack the blood cells of the fetus, and it can lead to death or it can lead to anemia. So we have to blood type the mother and the fetus. Well, we have to blood type the mother, and if she's Rh negative, then we have to administer a drug called Rogam to make sure she never has this happen. Okay. When you go through your simulation, you're going to see that they had a typo. It says the mother's immunological memory is now, it should say now, it says not. That's the opposite of what you want. So please correct that, it's now. So I went ahead and typed it again up here. The mother's immunological response is now set once she's been exposed to the Rh from the labor of the first child. Now she'll have the antibodies, and they can cross the placenta and attack the second baby if the second baby is Rh positive. So we are almost done. So how do you blood type a person? The easiest way is to use an Eldon card. Antibodies are already on the circles. So you have anti-A, anti-B, and anti-G. So the idea here is when you add a drop of the patient's blood to each of these circles, if the patient's blood has the antigen on their blood cell, it's going to react with the antibody on the paper and it will agglutinate. So you can see in this, the anti-A, there was no reaction. So this person does not have blood type A, but they did get agglutination on B. So they are blood type B. And here it says anti-D. You can see it clearly here, anti-D. This is what it really looks like in real life. This is what it looks like in your simulation. Same format though, anti-A, anti-B, anti-D. Anti-D is the Rh factor, remember. I wrote that somewhere on these slides. Oops, yeah, I wrote it here. Right, anti-D is the Rh, which is this. So this person is B positive because they agglutinated for B. That means their red blood cells have B antigen, and they also agglutinated with the Rh factor. So they have the Rh factor, so they are positive. They are B positive. Okay, you need to be able to identify all the different blood types, all the different combinations using this Eldon card. Okay, you're going to get some practice in the simulation. Make sure you actually do it. Take it serious. I also have a homework question, and you can bet that you will have a quiz question on that. Okay. Control doesn't have any and therefore the control should never have agglutination because there are no antibodies. If you get agglutination here, you know that your blood is contaminated, or the pipette or whatever utensil, right? So the whole test would be invalidated. You'd have to repeat the whole test if your control comes out clumpy. Okay. All right. So blood type O negative is the universal donor. O negative is the universal donor. Why? Because they do not have any antigens, red blood cell antigens. But you have to make sure it's O negative and not just O, right? O negative. Blood type A, B positive has every type of antigen. Therefore, any blood type is safe to give to them because they have all of the antigens. There's nothing you can give them that they don't already have. All right. So the idea here is that donors must not have an antigen that the receiver doesn't have, right? So the receiver has to have the antigen that you're giving them from the donor. Okay. So you could go through and make a list of all the different who can receive what blood types. But really, if you really understand these two principles right here, you should be able to solve any problem that you're asked. So if a person receives the wrong blood type, what's the big deal? Well, it is a big deal. Their immune system will respond, make antibodies, and then do their job and agglutinate all of the foreign red blood cells. And that agglutination is enough to block blood vessels and kill the person. So this is very serious. This is why we take blood transfusions, matching blood types so serious, because an immune reaction to a wrong type of blood can be fatal. Okay. And that's it.